All right. Hello, everyone. I'm Ed Ruthazer, and welcome to the first in the series of Canadian Neurophotonics platform Neuro Light Lunches. Thanks for coming. I just want to give a little bit of background here. So originally, this was part of a CAN satellite, a Canadian Association for Neuroscience satellite meeting that had to be canceled, unfortunately, because of COVID-19. And the satellite meeting was essentially intended to introduce some of the, up and coming researchers, new investigators, either from Canada or in Canada, doing neurophotonics based research or using neurophotonics in their research. And so it was a great opportunity to highlight some of the really exciting stuff that's going on. And we were disappointed when we had to cancel it. So this is our way to make that up to the community and offer a chance to hear some of these talks virtually. It's not as good, but maybe we can access more people this way. So that's the upside. Oh shoot, I have a honeybee that keeps coming into my room and he's found his way in. So if you hear me scream in the middle, I'll mute myself, but it's probably because I've been stung. So our speakers today, oh actually, and one thing that I wanted to say is that we have I'll put this up again. Too many tabs. Sorry about that. So this is again, the poster for the event. And I just want to draw your attention to the upcoming talks before we start today. So we've got 3 more sessions that are planned every Thursday at noon. And these will cover a range of topics from mini scopes and mesoscopic imaging to new exciting proteins that are being used for voltage detection, random access microscopy, development of new tools for looking at neuron-glia interactions. So the series of talks that we have are all really, I'm really quite excited about them, and I just hope that you'll add them to your calendar and come back next week as well. For today, our 2 speakers are Arjun Krishnaswamy and Tony Lim. Arjun is an assistant professor at McGill University. He's actually, he's been at McGill since 2017, I think, 2016, 17. He came from Harvard as a postdoc in the lab of Josh Sains, where he really started some of this exciting work on the retinal circuits. He also did his graduate training at McGill University with Ellis Cooper. And so I'm a little bit familiar with Arjun's work in that context. He's a really great speaker and a very thoughtful scientist. I think you're going to enjoy his talk. Arjun, I'll hand the platform off to you. Okay. One more thing I want to add is before Arjun starts is in order to avoid chaos, I'm going to be muting people, or please do mute yourselves if you haven't been muted. If you have questions to ask, in the participants tab, you can raise your hand. In addition, if you want, you can put something on the group chat. And when the time comes for questions, I'll be activating you one by one to ask the questions, okay? We won't have a huge amount of time because we've only got the full hour today, and I've already used a bit of the time. But Arjun, you've got 25 minutes or so for your talk, and then about 5 minutes for questions afterwards. So with no further ado, Arjun Krishnaswamy.
You can see my screen, right? It's all good?
Yes, it's excellent.
Okay, well, thanks, Ed, for that nice introduction. And thanks, Ed and Mario, for organizing all of this and inviting me. And thank you all for joining this Zoom call during these continuing weird times. I'm going to give you a short talk on just one of the stories going on in my new lab about the assembly of neural circuits and some optical tricks we've used to answer some questions. The lab is new, as you heard from Ed, and so as a consequence, I'm going to augment much of the story you'll see today with work I did as a postdoc with Josh Sains at Harvard. So on with the show. My general interest, my lab's interest, is to learn more about how the brain becomes wired and how its wiring, which is really the connections among neurons called circuits, give rise to a wide range of our abilities. So things that span from as simple as reflexes through sensation and motor control, perhaps all the way to more cognitive kinds of things. Now, the connections themselves are set down early in development and seem to revolve around the choices that neurons make. Choices about whom they should wire up with. But the way in which they go about making these decisions, the coordination needed to bring many neurons together, and indeed the consequences of some of these early wiring events for overall function are truly major open issues in the field and are issues that really motivate our work. And so as the title says, we try to gain some purchase on these problems by studying the circuits of the earliest step in the visual pathway of mice. And this is the mouse retina and its immediate targets in the brain. The reasons for this prep will become clear in a moment, but suffice to say, it offers us a number of advantages for our studies. The first advantage will be familiar. It's easily accessible because it's outside the skull, but it's in fact a part of your CNS, okay? It's a piece of your diencephalon that gets squeezed through the skull to form a thin sheet of neural tissue located on the back of the eye, onto which images of the world are focused by a lens, also within the eye. Now, if I were to take a cross-section through this structure and show you a cartoonified version of that cross-section, what you'd see is a really pleasing form of regularity. So you see all the neurons in this organ are arranged into three layers. There's an outer layer containing photoreceptors, an inner layer containing interneurons, and a ganglion cell layer which contains the so-named cells whose axons bundle together to form the optic nerve and send visual information off to the brain for further analysis. Now, some 60, 70 years of truly elegant studies has taught us an enormous amount about this organ and how the circuits within it are arranged and how they might work together to support vision. And so to just pull out a few facts germane to the talk, we know that every single one of these circuits begins with a photoreceptor. These guys are photon detectors. They're like the pixels in your phone cameras. Every single one of these circuits ends with a retinal ganglion cell, which, unlike the photoreceptors, come in a variety of flavors or cell types. And each of these types appears to be specialized to detect a particular aspect of the visual scene. So objects that move in a certain direction, edges with a certain orientation, colors, contrast, patterns, so on and so forth. Now, they detect their feature completely independent of their neighbors, but are arranged in parallel. And what this means is that if I were to label the pink cell and across the retinal surface, and on the next slide show you a real flat mount of the retina so we could see how it's distributed, you'd see that it represents itself evenly all across the retina's surface. And the thought is that it's trying to detect its visual feature from all points in visual space before sending it off to the brain. Now, all of the ganglion cells are doing this, and what this means is that the eye isn't a camera. Okay, it doesn't just take a picture of the world. Instead, the current thinking is that the eye would analyze a painting like this and decompose it into approximately 30 to 40 representations, with each of the images in this deck containing emphasis on a certain aspect of this painting, which in turn reflects the feature preference of one of these ganglion cells over here. And so we might imagine that one of these guys represents the low spatial frequencies in the image, sends a blurred version of the Mona Lisa upstairs. One that represents the green colors in the image. Perhaps if the painting were moving, there'd be one encoding the moving edges in the image, and so on and so forth. Now, about half of my lab is interested in the fate of this signal and the circuits downstream of the eye. I'm not going to talk much about that today. And the other half, and in fact the bulk of my work on this problem, has been situated on the first mystery, which is how on earth these ganglion cells even acquire these remarkable preferences. And so we know that it's nothing special about these neurons. It's nothing intrinsic to these cells. Rather, the thinking is that they're endowed with their feature preferences because of synapses they receive from a very specific mixture of interneuron types, of which there's about 120 or so interneurons, 45 different types of ganglion cells. So the kinds of things we're curious about are how does the pink cell find and wire specifically to the pink interneuron over here? What are the factors that direct that match? And given that they spend so much energy to wire up to one another, what is the contribution of this cell to that cell's feature preference? These are the kinds of things we'd like to learn more about in the next few years. And in the next few slides, I'm going to share with you a quick story about the role of cadherins in establishing this kind of matching pattern between these two cells over here. But before getting into that, I need to step back and tell you about the genetic approach we take to cracking these problems. And so if we wanted to learn how blue finds blue, or how it avoids green, or what the contribution of yellow is to the yellow ganglion cell, we'd ideally want to turn the retina into this cartoon, right? That is, we want a way to be able to mark and manipulate every single one of its neural elements. And without going into all the detail, it turns out that a sort of decade-long effort from small labs like mine and big consortiums like the Allen have yielded an encyclopedia of lines that allow one to do exactly this. And on the next slide, I'll show you a rogues gallery of some mouse lines we have in the lab, retinal cross-sections taken from them, with interneuron lines shown up top and ganglion cell lines shown down at the bottom. Now, there's a couple of things I want you to notice about these images. The retina is so stereotyped. You see these red lines that run through each of these images? These are the processes of an amacrine cell called a starburst amacrine cell. And we can mark them using antibodies to cholinacetyltransferase. And what it does is let us divide up the retinal neuropil, called the inner plexiform layer, into approximately 5 layers. So let me see if I can do this. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. The next thing I'd like you to notice is how each of the retinal types appears to confine its axons or its dendrites to just 1 or perhaps 2 of the 5 possible alternatives, which is an arrangement that led a fellow postdoc in Josh's lab, Shin Dwan, and I to wonder, well, could it be the way that you get specific wiring patterns is by these 2 cells, these 3 cells, knowing where to place their axons and dendrites? They recognize which layer in which to grow. If so, how would that kind of recognition even proceed? And so what Shin and I did was to get a hold of lines like this and seek out the expression of a selective expression of recognition molecules. And what we found was that these 2 lines up here that label bipolar cells, they have names. This is bipolar cell type 2 and bipolar 5. These 2 cells expressed a pair of highly related cadherins, with this cell, bipolar 2, expressing Cad8, and bipolar 5 expressing Cad9. Now, given that these 2 cells target almost mirror symmetric positions in the inner plexiform layer, and they are the only 2 cells that express these 2 isoforms over here, we wondered whether the cadherins are helping them reach these target destinations. And so what we did to address this idea was to get a hold of Cree lines, which grant access to these cells, and Cree-dependent reporters to reveal their anatomy, and we studied the consequences of what happens when you delete their endogenous cadherin. And when we did this, we found something really surprising, which is when we looked at bipolar 2 and we deleted its cadherin, 8, approximately half the cells stayed in the home layer, layer 2, and half of them moved off to a location that approximated where the type 5s live, okay? Deleting a cadherin that these neurons don't express produces no obvious effect, as we expect. Now, doing the same experiment in bipolar 5 produces a complementary deficit. Approximately half of these cells stay in their home layer, and half of them move off to a location that approximates where the type 2s live. These 2 neurons are more related than we initially thought. They seem to be making a mutually exclusive decision for these 2 layers in the inner plexiform layer, a decision that seems to require Cad8 in this neuron and Cad9 in that neuron. Now, given that requirement, we immediately wondered whether the cadherins are telling them where to go. And so to test this idea, what we did was to get a hold of these Cree lines and swap their cadherins. And what we learned was that it doesn't matter what bipolar cell type you are. What matters is what cadherin you express. Because we could take a bipolar 2, give it Cad9, push it down and make it look like a type 5 cell. Or we could take a bipolar 5, give it Cad8, and push it up and make it look like a type 2 cell. The cadherins are instructing these neurons to grow their axons into one of 2 locations in the inner plexiform layer, presumably so they can wire up to targets and create some of the circuits that we're talking about. And so what would these targets even be? It turns out that we got a really huge clue from these red lines, the processes of this starburst amacrine cell I mentioned a few slides ago. This cell is rather famous in the retina literature because it provides inhibition to a ganglion cell called the direction-selective ganglion cell. And it has dendrites that look like this. They're bistratified and they grow directly into layers 4 and 2, exactly where these 2 guys go. Now, given that we effectively have 4 neurons here that target 2 layers, and we know exactly why 2 of them show up in the first place, we immediately wondered whether these neurons create a circuit together. And so to determine whether this was true and indeed to have a general strategy for mapping out connections amongst retinal neurons, mapping out circuits, I devised the following approach. What we do is get a hold of mouse lines that put GFP in a particular type of ganglion cell, like this. We then cross those lines off to ones that put the enzyme 3 recombinase in interneurons, like that. Then, either by way of a mouse cross or by way of a virus, we can install the light-sensitive ion channel, channel rhodopsin 2, into the interneuron's membranes, record from the ganglion cell using whole cell patch clamp methods, activate the channel rhodopsin, and then use synaptic transmission as our readout of connectivity. Now, without going into tons of detail, this is a difficult thing to do because channel rhodopsin is excited by light, and here sit the photoreceptors. And so we had to devise some methods to activate the opsin using 2-photon excitation, which sort of creates a spot, kind of like my fake laser pointer here, which we could swing around the neuron and then drive it to create transmission in this postsynaptic cell. And without going into tons of detail, this at the time was a technically challenging task. And so I had to learn how to build a microscope that could do this kind of experiment. And I'm happy to talk more about the details of this thing afterwards if you're curious. What I'd like to do now is to bring you into an experiment and show you the kinds of data that we get from this trick. And so on the next slide, what you'll see are the positions of a ganglion cell and a field of interneurons, bipolars, that I'm stimulating. They live in 2 different layers, but here I'm just showing you their planar position. So I'm patching onto this guy, and we're going to stimulate each of these gray dots, let's say 5 or 6 times, collect the responses here, average them, and throw them back in the location of the cell that we stimulated. Doing that for all these cells creates a heat map of connectivity. And what you can see is that this neuron receives its input with a very pronounced tilt. Now, there's a couple of things we were happy to see. These are whole cell currents. We're quite zoomed out here, but they're inward pointing. They have linear IVs. They're sensitive to glutamatergic blockers. That's a really good sign because bipolar cells form glutamatergic synapses. The other thing that comforted us was the tilt, because these ganglion cells grow dendrites with a pronounced tilt. So they can really only collect input from this side of the cell. And so now we have a trick to map out connections between retinal types. And so what about our problem? What about our question? Do the cells connect to one another? Well, in the case of bipolar 2, yes, they do. In the case of bipolar 5, the same is true. We showed that bipolar 5 provides excitation to starburst amacrine cells, and we confirmed an older result showing that starburst amacrines provide inhibition to the ganglion cell. The 4 neurons have this particular synaptic relationship with one another, and we know why 2 of them show up there in the first place. So what's going to happen? What's going to happen when we go in and delete CAD9 in this cell? Well, it's not entirely clear, right? Approximately half the cells stay and half the cells move off to this layer over here, seemingly well within synaptic striking distance of this ganglion cell's other dendritic arbor. Maybe nothing would happen. And so you can imagine our surprise when we went in and did the experiment and found that although in controls, approximately half the bipolar cells within the sort of dendritic capture of the ganglion cell provide input, the loss of these cadherins over here reduce this to around 10% or so. So pretty severe reduction in excitatory drive to this cell. And I simply don't have time to talk about the consequences of these mutations for the visual responses of this neuron, but effectively you end up ruining its light responses. And I can tell you more about that afterwards if you'd like. What I'd like to do instead right now is to zoom out a little bit and talk a bit more about cadherins in the context of all 4 cells a bit more. So let's review a little bit of what I've said. I've told you about CAD8 and 9, which guide these 2 cells to these 2 layers, presumably so they can synapse up with red and green over here. All the cells have names, and we know their anatomies quite well. And we have really great genetic access to these neurons over here. But you know, there was something that always bothered us about this story, which is that I haven't told you anything about how the cadherins guide them to those layers. Presumably, it's through their adhesive interactions. Problem is that cadherins like to bind to themselves across the cell-cell junction, and these are the only cells that express CAD8 and CAD9. it would turn out that a couple of parallel observations would come to our rescue. The first came out of some genetic and sequencing studies going on in Josh's lab, which showed that the postsynaptic neurons express CAD6 and 10. And the second was an observation from the literature showing that cadherins can in fact bind heterophilically. They just like to do so to closely related isoforms. And it just turns out that the closest related isoforms to CAD9 are in fact CAD6 and 10. And so it was this kind of arrangement that led Shin and I to wonder, well, maybe it's some overlapping adhesive interaction between these three isoforms across the three cells that help them wire up to one another. Now, addressing this would require some double and triple knockouts, which is a technically demanding task, and that's because the three isoforms live back to back on chromosome 15, within a recombination distance, and so Shin employed CRISPR-based genome editing methods to generate the required knockouts. And on the next slide, I'll just show you some data from the triple knockouts. The doubles don't matter because of compensation issues. And what we found was quite interesting. When we deleted 6, 9, and 10, now the ganglion cells seemingly didn't know where the layers were and grew these scrambled looking dendrites, which you can see from these line scans over here. Interestingly, the starburst amacrins didn't care at all. They didn't seem to care whether we removed CAD6. And we were interested by this because these neurons are amongst the first born in the retina and amongst the first to laminate in the inner plexiform layer, which led us to wonder, well, maybe these guys are the first ones to come in, and mechanisms related to this kind of overlapping cadherin interaction help position these cells and possibly other ones. And so to test this notion, what we did was to get a hold of chat-CRE, install a receptor for diphtheria toxin into this cell over here, applied the toxin very early in development, and destroyed these cells. And what we found were phenotypes that were remarkably similar to what happens when you delete 6, 9, and 10. Taking these two guys together, it makes you think that maybe the ganglion cell, without these recognition proteins, simply can't see those layers anymore and ends up growing these scrambled dendrites over here. And so going forward, we're trying to extend this sort of theme that maybe the cadherins are specialized to direct these kinds of layer targeting events. If so, I suppose a simplistic model would require every layer to have a cadherin. Seems sort of silly in some ways. Perhaps a little too simple because you see there's too many neurons, right? There's 120 types of interneurons and there's 45 types of ganglion cells. It's got to be the case that there are many different kinds of feature detectors with different sorts of connectivity that live within the same layer. And without going into tons of detail, we have some stories in the lab that suggest a different gene family called the immunoglobulin superfamily is responsible for keeping the connectivity of these two cells apart. What I'd like to draw your attention to is the division of labor. If IGs are for targets and CADs are for layers, it sets up a sort of cute speculative scenario in which you could mix and match a limited number of tags from the two families to potentially wire up this kind of diversity over here. And this is an idea that we're pursuing in a couple of ways in the lab. I'll share with you one on the next slide. I should just say that reality isn't terribly far off of this kind of diagram here. We do have great candidates for the different layers. And I've done some work on a couple of IGs called sidekicks and their role in local synapse formation. And so in my final 4 or 5 slides, I'd like to share with you some data from a graduate student in my lab, Aline Rangel Olguin, who's been working on cadherin 4, also known as retina cadherin. And the reasons for this is because it's fairly understudied and because it contains 2 neurons that are sidekick positive, setting up an eventual experiment where we delete it and ask what happens to the mistargeted cells. We could ask whether they rewire to nearby IG match targets. But let's just see her data on generalizing the role of cadherins over to this isoform. On the next slide, you'll see some antibody staining of CAD4. And what we find is that it's sort of in the upper parts of sublamina 3 and in the top parts of sublamina 1. This corresponds to the morphology of the cells that express CAD4, which we can label using a line we have in the lab, which is a CRE-ER knock-in to the CAD4 gene. Crossing to a reporter reveals a variety of ganglion cells and some sparse interneurons as well. The ganglion cells do the same thing as the protein, so it's probably on the dendrites, going to upper S3 and into S1, There's many types of cells in here. In fact, in some dense tamoxifen-treated retina, we estimate that it's somewhere in the ballpark of 1/4 of all ganglion cells, so it's an awful lot. And what Aline's been doing has been to kind of look at the consequences of CAD4 loss from both functional and anatomical points of view. And the reason for the function is because of the staining pattern of CAD4. You see, all the cells in the retina that respond to light offset layer in the CAD4 positive layers. All the ones that respond to onset are CAD4 negative. And so what she's wondering is, if I delete CAD4 and cause these neurons to wander off, I should at minimum deplete the number of off cells I observe when I do population calcium imaging in the retina, and perhaps see some that have converted into on cells. And so let me show you how she does the experiment. She takes a pan-RGC line, infects the retinas with Cre-dependent GCAMP6F, allows her to image across many fields, thousands of neurons. She's trained a machine classifier to go in and circle cells for her, so we can do that in an unbiased way. And then we can pull out the responses of these cells to a variety of stimuli. I'll share with you one called a full field flash, which is like it sounds, where we make a screen go dark, bright, dark, and gray, and we can pull out the great families of ganglion cells. On cells, you can see off cells and on-off cells. And this is sort of seen a little bit more clearly on the next slide, where I convert the traces to an image. So here, every column is a trace from a single neuron. And Aline's found a way to cluster the off cells to this side, the on cells to that side, and down here in the middle are the on-off cells. Okay? And she does this by computing an on-off index. All right? And I don't have time to tell you about it, but ask me about it afterwards. Showing you the histogram of the on-off index score shows the same thing: off, on-off, and on. What's cool is when she goes in and does the CAD4 knockout, we suddenly deplete from the off column and we gain in the on column, suggesting that the cells are mislaminating and potentially rewiring. And in a limited number of single cell tracing cases, that's in fact what she finds. So ganglion cells that ought to go up here are now showing these kind of disordered dendrites, very reminiscent of the direction selective cells from the first part of the talk in the absence of 6, 9, and 10. And so some future directions involve determining, well, are these in fact the same cell that are changing in the absence of CAD4? It's increasingly easy these days due to a nearly complete molecular taxonomy of the cell types in the retina. So we can image, apply markers, and figure out which cells are which, and determine whether responses like that are normal or due to some miswiring event. The same trick applies over here. And we're in the process of building a new kind of microscope that will allow one to apply the optogenetic stimuli from the first part of the talk with the dense calcium imaging from the second part of the talk to rapidly map out connections in the retina. And so that's sort of the future, but I think for the present, I should stop and thank all the people who helped. My advisor, Josh Sains, my friend Shin Duan, who's now at UCSF, important members of the Sains Lab, my lab at McGill. You saw a lot of Aline's work today, some funding, and a long-term collaborator, Cook, and all of you for your attention. Thanks very much.
Thank you so much, Arjun. That was a fantastic talk. So if you have questions for Arjun, please go to the participants tab and raise your hand. Right now I see a lot of applause. No questions showing up.
Someone
doing it wrong.
Someone named Casper.
Casper Pogorski. So Casper, are you able to unmute yourself? I don't know.
Sure. There might be a vacuum in the background. I'm sorry.
Never apologize for cleaning.
Can you hear me?
Sorry.
Yeah.
Go ahead.
It was a beautiful talk. I was really curious. You said that when you delete a particular cadherin, the cells will branch in one or another of these sublaminae. What keeps the processes within a single layer or sublayer?
Yeah, you mean like in the absence of the CAD, what's preventing them from growing all over the place?
Or branching into both layers at the same time, for example.
It's a good question. Yeah, I guess in the bipolar case, I remember, in the bipolar case, we did some single cell reconstructions, and we know that those guys don't create two branches. As to whether the ganglion cells, particularly at the end, with this cadherin 4 mutant, we actually think that what you're saying is probably happening. So we don't often see cells that are bistratified in our controls, but in our knockouts, we are seeing some that are bistratified. And so could it be that CAD4 also has some role in like dendritogenesis or something like that? It's also possible. At least for the first part of the talk, I don't think that's the case. Now, as to why they default to 2 locations or 3 pre-specified locations, your guess is as good as mine. There are mechanisms that occur slightly before this layer matching process, which kind of restrict cells to the off domain or to the on domain, if you know what I mean, and some other things like that, things like semaphorins and some other stuff. So those sorts of mechanisms still operate, and so maybe we're just unmasking a previous developmental state or something like that. Not sure if that answered your question, but I kind of discussed it a bit, I guess.
Yeah, absolutely. Thanks.
Okay, are there any other questions? I don't seem to be able to see anything on the list of attendees. Here we go. Posting them on the Zoom chat is the best way. So Salia, would you like? Go ahead and ask.
Yeah, so I did my undergraduate thesis in like computational imaging microscopy. So I guess I'm interested in what you think is a limiting technology for your experiment. Like, do you find the imaging technology that you're using provide sufficient resolution to, you know, do your analysis? Do you find the image processing software and algorithms are like filling all your needs, or what is the current limitations in that aspect of your technology?
Yeah, it's a good point. So in terms of the kind of fixed section, anatomical tracing, that sort of stuff, I would say that we have the same kinds of challenges you would find anywhere else in the brain, especially when it comes to fine processes and determining, you know, truly who's connected to who. And I mean, my entire talk was about functional connectivity, right? I mean, the synapses could in fact still be there, but just silent in some way, right? I didn't show you any evidence that would argue one way or another. I would say that the thing that we're trying to do at the moment, which we feel would be really, really helpful, is that at the moment in the retina, we kind of, there's a feeling that we'll know all the cell types. And that's because of some new sequencing tricks that are out there. There's really great genetic access. And so what remains is to stitch them together into these circuit type patterns. And so we're hoping to address that with this all optical method. It's quite difficult to do. So I suppose if you were looking for things to improve upon for the community, it would be to make that sort of technology more accessible. I don't know. Do you have something in mind? It feels like maybe you thought of something.
I guess when I was developing the algorithms, I mean, I was more interested in trying to make the image reconstruction time as close to real time as possible. So you could
through a stack or something like that. Is that what you're thinking?
Yeah, like just trying to, because you want to observe the functioning of the neurons in real time. So like you want to, I mean, usually there's a trade-off between what resolution you can get and how quickly you can get the image reconstructed. So that's what I was interested in. But you said what could be useful is developing a technology that would identify all the retinal cell types.
No, so I think that kind of thing exists. I was thinking of faster ways to map out connections between the two. So if you had a really great optical trick to somehow sort out a bunch of reporter labeled cells and say, oh yeah, it's this one, this one, this one, this one, this one, this one, that would be terrific.
Real time.
Okay, faster ways to identify the connections between cell types. Okay.
Yeah, thank you for that question, Stella. I think maybe Jennifer has one question. There's time for one more. Jennifer, did you want to ask your question?
Sure. Hi, Arjun. That's a very nice talk. Maybe this is for offline discussion as well. Can you hear me okay?
Yes.
Okay. Yeah, so more and more these days, I'm actually questioning more what's happening in the retina during development before I even start to think about cortex or even colliculus. So you mentioned this just very briefly, maybe this mistargeting reflects some normal developmental stage. Do you, should I be thinking about this super early developmentally, or maybe possibly later postnatally?
Oh goodness,
Maybe we should discuss.
I suppose that just and it's a bias, I'll admit it. I don't often think about the steps following, kind of activity dependent things in the retina, because there is sort of this bias in the field that there isn't all that much in the way of role for activity and the kind of layering patterns I talked about today.
Sure.
I mean, there's beautiful work from Rachel Wong's group about
Sure.
the kinds of connectivity changes in the planar geometry of the retina. In terms of earlier things, what I was thinking about was, like Jeremy Kay's work, like all the beautiful work from Alex Klodkin, that as the cells are growing in, there is a tendency for them to want to grow into certain layers and then perhaps, or into certain regions, and then they sort of sort themselves out. It's that latter sorting that I think that I would probably say the CADs are useful for. Maybe Josh would say the same thing, Josh and Shin. But there are earlier mechanisms that kind of predispose neurons to, let's say, choose the upper half or choose the bottom half of the IPL.
Yeah, no, interesting. I was just thinking more in terms of like maybe a developmental shift, even postnatally, of like on-off responses, and if that's something that might actually come from refinement, even all the way probably at the retina. Sorry. Anyway, something maybe fun to think about.
Yeah, absolutely. Yeah, I mean,
or even evolutionarily.
Yeah, let's talk about. I mean, I don't know.
Maybe in the interest of time, we should.
Sorry. Yeah.
Okay.
No,
I didn't mean to cut you off. Thank you for participating today, Jennifer. I'm happy to have you here. I think you're the Jennifer. I know you.
Yeah, I know you.
Okay, good, cool. Thanks. Nice to see you again.
Yeah, you don't need to see my house.
Exactly. Or your last name.
All right. Yeah, let's hear from Tony. Sorry, Arjun. Talk about later.
So thank you so much. Please, everyone, join me in thanking Arjun. It was a really fantastic talk, and as always, if you want to take conversations offline, that's always welcomed. Thanks again.
Yep. Thanks for the invitation. Thanks, guys. Yeah.
All right. So our next speaker is going to be Tony Lim. Tony Lim is actually a postdoctoral fellow in my laboratory. And he's been working on microglial trogocytosis in the developing visual system. Tony actually did his PhD at McGill University with Zhejiang. So he comes from an immune system background, and he worked in pain previously. And so now he's brought his expertise to study this topic, which has become quite important and quite interesting in the developing visual system. And so Tony's talk today will be microglial trogocytosis and the complement system regulate axonal pruning in vivo. Tony, I'll hand over the podium to you.
Thanks, Ed. Thanks for organizing Ed and Mario, and for the wonderful introduction. Okay. So I'm very excited to share this story that Ed and I have been working on. It's titled Microglial Trogocytosis and the Complement System Regulate Axonal Pruning in Vivo. So microglia are thought to contribute to developmental synaptic remodeling by removing inappropriate synapses. For example, in mammals, in the lateral genetic nucleus, the retina projects. So the lateral genetic nucleus receives inputs from both eyes. And at birth, the axons from both eyes overlap, but during the first postnatal week, axonal pruning causes a segregation into eye-specific territories with minimal overlap. So this process is important for binocular vision. And this is what the segregation looks like in a wild-type mouse. Using different retrograde tracers, the contralateral and ipsilateral retinal ganglion cell axon targets can be mapped. And as you can see here, there's minimal overlap between the different eyes. When microglial disruption, when microglial function is disrupted, the segregation into eye-specific axonal regions fails to occur, leaving substantial overlap between the ipsilateral and contralateral axons, demonstrating that microglia are important for the removal of inappropriate axonal projections. So the mechanism by which microglia are thought to eliminate synapses is through phagocytosis. There's a lot of indirect evidence to support this. So for example, immunohistochemistry studies have shown that microglia contain presynaptic components within them, for example, SNAP25, as well as postsynaptic markers, PSD95, for example. And then this is also confirmed by electron microscopy studies, which have found dendritic spines, as well as axon terminals within microglia. So how do microglia remove synapses? So the mechanism by which microglia eliminate synapses has recently been shown to be through trogocytosis, or a partial elimination. So trogo means to gnaw or nibble, and it seems that microglia are nipping off small pieces off of axons and synapses. So this is work from ex vivo slice culture. So in teal are presynaptic puncta, and in red is a microglial cell. And sorry, this video is not going to play. So here you can see that this presynaptic puncta here is kind of pinched off by the microglial cell and pulled away. So again, this work was done ex vivo, and it remains to be seen whether or not it occurs in vivo. And so in the work that I'm about to present, we will show you that trogocytosis by microglia does occur in vivo. And to do this, we use the developing Xenopus laevis retinal tactile circuit. Then we show that microglia regulate axonal pruning in the circuit, and that microglia are also important for the proper wiring and function of this circuit. Finally, we will then examine the molecular mechanisms that regulate microglial-mediated axonal pruning at the single axon level. So this work was carried out using Xenopus laevis tadpoles. So for those of you that are unfamiliar with the model, this is what they look like. The albino animals are highly transparent, and their brain is located at the dorsal surface of the animal, as you can see here, making them highly amenable for in vivo imaging. The retinal tactile circuit of these animals can be used as a model to investigate neuronal circuit formation. In this circuit, the retinal ganglion cell axons project to the contralateral optic tectum, where they synapse on tactile cell dendritic arbors in a region called the neuropil. And and we can label the we can label the retinal ganglion cell axons by electroporating the retina with plasmids encoding fluorescent proteins. For example, in this case, is EGFP. And you can also label microglia by injecting IB4 isolectin conjugated fluorophores, in this case, IB4 isolectin alpha 594. And we can see these highly dynamic cells, which are microglial cells. And to our knowledge, this is the first in vivo imaging study of microglia in Xenopus laevis. So just to show that IB4 isolectin does indeed label microglial cells, we performed a laser radiation injury to the neuropil. And as you can see, the IB4 isolectin labeled cells mobilize to the injury site and phagocytose injured tissues, providing functional confirmation that these are microglial cells. In zebrafish, it's known that microglia do not associate with the neuropil, whereas in mammals, microglia are often associated with neuropil regions. We wanted to investigate whether or not microglia associate with neuropil in Xenopus. So to do this, we labeled the neuropil region by bulk electroporation of retinal ganglion cells to label as many retinal ganglion cell axons as possible with a GFP. And we also labeled microglia, and then we did some time-lapse imaging, and we found that, yes, the microglial cells do appear to interact with the neuropil in Xenopus laevis. So now we're going to take a closer look at this interaction. So in these time-lapse images, a single optical section is shown. And we can see that the microglia are not excluded from the neuropil region. They can, for example, in this example here, you have the microglia entering into the neuropil region from outside of the neuropil region. And also the microglia are freely able to move through the neuropil region. So the neuropil does not exclude the microglia from entering into the neuropil. And also microglia can be observed extending processes into the neuropil to contact axons. Here's some examples of that. So the microglia are contacting axons. Is this mediating trogocytosis, for example? We wanted to investigate that, and to do that, we labeled retinal ganglion cell axons with pH-stable GFP, also called pH-TD GFP. So that's here in green, and we labeled microglia with IB4 isolectin, which we show in red. And the co-localization of red and green is shown as white. So if we look at this microglial cell here, it's interacting with this axon. And after the interaction, it becomes more fluorescent green. Here's another example. So have a look at this microglial cell. It's going to interact with the axon there. And you can see that after interaction, it becomes more fluorescent green, like in the previous example. So in both cases, so these are two examples of direct in vivo evidence of trogocytosis of axons by microglia. So when a microglial cell trogocytoses a pH-stable GFP-labeled axons, it increases in green fluorescence. Since microglial cells are constantly moving around in the developing Xenopus laevis tadpole, they would need to be followed constantly in order to measure trogocytosis in an individual microglia. So that's complicated because they might leave the imaging field, resulting in a loss of tracking. In order to quantify trogocytosis, we decided to look at the green fluorescence over the population of microglia within the optic tectum. So if the number of labeled axons in the optic tectum increases, we can expect that the frequency of trogocytotic events between microglia and labeled axons to also increase. And if that happens, then we should see an increase in the green fluorescence in the population of microglia, which we would then measure as a proxy for microglial trogocytosis. So using this principle, we developed an assay to measure microglial trogocytosis of axons. So in this assay, retinal ganglion cells were electroporated with pH-stable GFP, and also microglia were labeled with IB4 isolectin at developmental stage 39 or 40. And then 2 days after electroporation, the labeled axons begin expressing pH-stable GFP and begin to innervate the optic tectum. And at the same time, the microglia are arriving to colonize the optic tectum. And we then do in vivo imaging at day 4 and day 5, where we count the number of microglial cells, and we count the number of pH-stable labeled axons, pH-stable GFP labeled axons within the optic tectum, and also measure the green fluorescence within the optic tectum, within the microglia within the optic tectum. Okay, so here's an example Z stack with 2 axons, and using the microglial channel in red, we can create 3D mass to isolate the green fluorescence within the microglial population. So like so. And it's important to note that when, so then with using this method, we can get the green fluorescence within the microglial population. And it's important to note that when apoptosis was detected, either by the presence of apoptotic bodies or by reduction in the axon count, then the data was excluded, just to avoid the confound of apoptosis. So when we electroporate, the number of labeled axons is random. Oftentimes we get no labeled axons. And when there are no labeled axons in the optic tectum, we see no change in the green fluorescence in microglia from day 4 to 5. And you might be wondering why microglia are fluorescent green at all, since there are no labeled axons to interact with. And that's because microglia are myeloid cells. And one property of myeloid cells is that they are autofluorescent. And when one or more labeled axons are present in the optic tectum, we see that microglia are significantly more fluorescent green on day 5 when compared to day 4. So this suggests that microglia are collecting green fluorescence from the axons from day 4 to day 5, which we use as an indirect measure of trogocytosis. Next, we wanted to know whether or not microglial trogocytosis of axon plays a functional role. After all, it's entirely possible that trogocytosis has no effect on circuit connectivity or development. And in order to answer this question, as a starting point, we used a very crude method of just depleting microglia. If there are fewer microglia, then there should be less trogocytosis. So to deplete microglia, we used a drug called PLX5622, which is an antagonist for the colony-stimulating factor 1 receptor. Without colony-stimulating factor 1 receptor activity, microglia cells undergo apoptosis. So Thus, we see a reduction in microglia numbers when the animals are reared in a solution containing PLX5622, as you can see here. So now we can ask the question, what is the effect of microglial depletion on axon morphology? We treated animals with vehicle or PLX5622 and electroporated retinal ganglion cell neurons with pH stable GFP, and we followed the axons over several days and quantified axon arbor size and branch numbers. We found no difference in axon arbor size when microglia were depleted, but we did find an increase in the number of branches per axon when microglia were depleted. We then asked what was the effect of increased retinal ganglion cell axon branching on the function of the retinal cortical circuit. So to do this, we developed a simple and robust behavioral test where we used a modified projector to present visual stimuli to a tadpole, and then recorded the behavior from above with a webcam. So this is what the setup looks like in reality. It's a very inexpensive setup. With our current setup, we have an $80 projector, which projects to a screen, which is just a white piece of paper, and this is an $80 webcam. The most expensive thing required is a laptop with Python installed. And we then presented dark looming stimuli, which simulates a oncoming, oh, it's not working. Okay, well, anyways, it's a growing black disc is the dark looming stimuli, and that simulates an oncoming object or predator. And this sort of stimuli is common for eliciting escape behavior in highly predated animals. And as a second stimulus, we presented bright looming stimuli. So bright looming stimuli do not correlate to a stimulus in nature, which we would expect tadpoles to react to, and is a relatively poor stimulus for eliciting escape behavior. So again, the dark looming stimuli is a growing black disc on a white background, and the bright looming stimuli is a growing white disc on a black background. So we'll first talk about the response to dark looming stimuli. In vehicle-treated animals, why is it not working? Okay, hold on. Let's just, I changed the, does this work? Okay, so in vehicle-treated animals, we see that the dark looming stimuli elicits stereotypical escape behavior, which we can detect with contrail. And the contrails of 10 animals are shown here. And with microglial depleted animals, the dark looming stimuli does not seem to elicit escape behavior. And here's our contrails of 10 trials. And if we And here the looming stimulus is applied at time 0, and we're looking at the instantaneous velocity. And we see that in blue, we have the vehicle-treated animals. And you can see there's an increase in velocity, instantaneous velocity, whereas microglial depleted animals do not respond to the looming stimulus with an increase in instantaneous velocity. And this is, we can quantify the distance traveled before and after the looming stimulus, and after looming stimulus, vehicle-treated animals respond to, by significantly increasing in distance traveled after the dark looming stimulus. Whereas this is not observed in microglial depleted animals. Whereas we also looked at escape probability, and escape probability was significantly reduced by microglial depletion. And when the tadpole did make escape movements, the escape distance was not significantly altered by microglial depletion, suggesting that microglial depletion does not alter motor responses. So now we'll talk about the response to bright looming stimuli. And so here's an example response to bright looming stimuli. You can see that the tadpole didn't really respond. This is a vehicle control treated tadpole. Whereas microglial depleted tadpoles have produce a robust escape response to the bright looming stimuli. And when the dark luminous stimulus is presented here at time 0, it causes an increase in the velocity of microglia depleted tadpoles, seen here in red, but this increase is not seen in microglia depleted or in vehicle treated tadpoles. Sorry. So microglia depletion increases the distance traveled to bright looming stimuli. Whereas, and in addition, microglia depletion increases the escape probability to bright looming stimuli. And, but again, these results are not due to changes in motor function, because when the tadpoles do make escape responses, there is no significant increase in the escape distance. So the results support this model where in normal development, microglia-mediated axonal pruning reduces axonal arborization, resulting in appropriate wiring and appropriate behavior to looming stimuli. Conversely, in the case of microglia depletion, axonal arborization is increased due to impaired microglial-mediated axonal pruning. And then this results in inappropriate wiring and inappropriate behavior to looming stimuli. So clearly microglia are playing some role in the proper function of the retinotactile circuit. And while we observe that microglia are trogocytosing axons, it's still unclear whether the effects of microglia depletion on axonal pruning are occurring through trogocytotic mechanisms. So for example, it's known that microglia cells secrete trophic factors and cytokines, which can affect circuit function. Therefore, it's possible that the effects of microglial depletion are due to non-trogocytotic mechanisms. So what we need is a tool to inhibit trogocytosis without affecting microglia function. So to find such a tool, we started by looking at molecular mechanisms that regulate microglia-mediated synaptic pruning. In the literature, the molecule that has been most often implicated as a key regulator of microglia-mediated synaptic pruning is complement C3. So C3 is a fluid phase complement protein which exists at high levels in the blood and CSF, and it's hypothesized that C3 tags inappropriate synapses that are to be removed by microglial cells. As, for example, microglia are known to have receptors for C3, in this, for example, complement receptor 3, and the binding of complement receptor 3 to C3 induces phagocytosis. And it has been hypothesized that appropriate synapses that should not be removed by microglia, they express a complement inhibitory molecule to protect the synapse from C3 tagging. The identity and existence of such a molecule that is endogenous to neurons is unknown, but we decided to look for one in Xenopus. And so we carried out a bioinformatics search to find such a molecule, where we queried complement C3 on a protein interaction database, which provided us a list of proteins which are known to interact with complement C3. These red proteins are proteins which have complement control protein domains within them, which are known to negatively regulate the complement system. So we then use these proteins that we got, these negative regulators of complement activity, and search them on a transcriptomic data set to see whether or not neurons would express these complement inhibitory protein molecules. So the neurons are the orange and purple data sets, cell types in this data set, and you can see that only CD46 is highly expressed by neurons. So CD46 is a well-characterized complement inhibitory molecule, which is known to inactivate complement C3. So that sounds like a promising lead. And so we searched the Xenopus for the Xenopus homolog of CD46 using BLAST. And the most similar protein was amphibian regulator of complement activation 3. And so we decided to model CD46 and ARCA3. And you can see that it looks very similar. They have very similar structures. In both cases, the extracellular part of the molecule looks like sausages on a string. And it's known that in CD46, the bottom two complement control modules bind and deactivate complement C3. And CD46 has 4 complement control modules, whereas ARCA3 has 8 such modules. So both proteins are anchored to the surface of the cell membrane near the C-terminus with a transmembrane region, and both proteins also have a short intercellular tail. So then we modeled, so then we wanted to see what, whether or not ARCA3 is expressed in retinal ganglion cells. And to do this, we used RNA-scope, in situ hybridization on retinal tissue, counterstained with Herxt in blue. And so the negative control probe we don't see any transcripts in the retina as expected, and also as expected, the positive control probe, which is RNA polymerase 2, a housekeeping gene, identified transcripts ubiquitously across the retina. The probe for ARCA3 identified transcripts concentrated in specific layers of the retina. In particular, transcripts were concentrated at the ganglion cell layer, which is where the retinal ganglion cells are, demonstrating that ARCA3 is endogenously expressed by retinal ganglion cells. We then wanted to know whether or not ARCA3 localizes to the correct cellular compartment to protect synapses from the complement system. And to do this, we created a fusion protein where ARCA3 was tagged with M cherry. And this was then co-expressed with synaptophysin pH stable GFP. So synaptophysin distributes to synaptic vesicles and is a marker for synaptic puncta. And when we look at the distribution pattern of ARCA3, it has a very similar pattern to that of synaptophysin. So the two are highly colocalized. And when we do a colocalization analysis, we find that ARCA3 and synaptophysin pixel intensities are highly correlated, suggesting that ARCA3 distributes to the same cellular compartment as synaptophysin, which we know is a presynaptic marker. So ARCA3 distributes to the correct cellular compartment to protect axons from trogocytosis. Now we can use our trogocytosis assay to determine whether or not an overexpression of ARCA3 would inhibit trogocytosis by microglia. So again, when one or more pH stable GFP labeled axon is present in the optic tectum, the microglia become more fluorescent green on day 5 when compared to day 4 after electroporation. But when we also express ARCA3 and overexpress it in the retinal ganglion cell axons with pH stable GFP, we do not see a corresponding increase in microglial green fluorescence when pH stable GFP labeled axons are present in the optic tectum. So this suggests that ARCA3 overexpression is inhibiting microglial trogocytosis. So next we examined the effect of ARCA3 overexpression on axon morphology. In this axon is overexpressing ARCA3, and this is the control axon. We found that when ARCA3 is overexpressed, axon arbor size is significantly increased. And in addition, ARCA3 overexpression also significantly increases the number of branches. So if the effects of ARCA3 on axon morphology are mediated by inhibiting the complement system, we would expect that by enhancing the complement system, this would produce the opposite effect. And to test this, we created a VAMP2-C3 fusion protein. So VAMP2, or synaptopreverin, is a synaptic vesicle associated protein which distributes to the axon surface. And so this VAMP2-C3 fusion protein tags C3 to the surface of the axon. And as an additional control, we also created a construct where we just overexpress VAMP2 to ensure that the effects of VAMP2-C3 were not mediated by an overexpression of VAMP2. And we can see that expression of VAMP2-C3 in retinal ganglion cell axons significantly reduces axon arbor size when compared to both control or VAMP2 overexpression, and also reduces branch numbers when compared to control or VAMP2 overexpressing axons. So what we found was that the complement activity modulates axonal pruning by inhibiting the complement system at the single axon level. This results in an impaired axonal pruning. On the other hand, if we enhance complement activity at the single axon level, this results in excessive axonal pruning. So to summarize, using the Xenopus laevis retinal tectal circuit, we observed direct in vivo real-time evidence of trogocytosis of axons by microglia. In addition, we found that microglia regulate axonal pruning in the Xenopus retinal tectal circuit. And we also found that microglia are important for the proper wiring of the circuit and for appropriate behavioral responses to looming stimuli. Finally, we found that the complement system regulates axonal pruning by microglia at the single axon level. So this work wouldn't be possible without the guidance and support of my supervisor, Ed Ruthazer, who has been an amazing mentor and source of inspiration for me to look up to. And I'd also like to thank the members of the Ruthazer Lab for the happy and caring work environment, as well as financial support from CIHR. Thank you very much for your attention.
Thank you very much, Tony. That was great. Are there any questions from the audience? And again, I think the best thing is to just type something in the group chat, because that seems to be the thing that shows up most readily. Any questions for Tony? Here we go. So Mariev, would you like to ask your question?
Yes, just a second. I was just wondering if your all your work is done with the intact C3 molecule, not with the cleaved C3?
Yes, so it's for example, the VAMP2 C3 construct, is that what you're getting at?
Because typically the C3, this intact C3 is not doing much, right? You require like activation of C3.
Right.
So I was just wondering.
Right. Yeah. So we in our construct, the VAMP2 C3 construct, it is the full-length C3 molecule that that I created a fusion protein with VAMP2, and C3 is known to undergo spontaneous hydrolysis to self-activate through the alternative pathway. So I think when we tag the axon with VAMP2 C3, the C3 can then spontaneously hydrolyze and then activate.
Okay.
Yeah.
So it wouldn't be enough to like trigger a real, downstream events of the immune system, but just enough for this.
Yeah, I see. Yeah, I think it could, but I'm not sure. I don't have that much experience with the molecule in terms of like, I didn't quantify, for example, whether or not like cells that express more of this VAMP2 C3 construct are more likely to get undergo apoptosis or be phagocytosed completely. But that's a potential possibility, I think.
Okay,
yeah.
Cool. Thank you.
Thank you for your question.
Okay, there's a question from my old friend Yajing. Do you want to?
Yeah, hi.
Hi.
Thanks so
Hi,
much for the
Yajing.
talk, Tony. I was just wondering, because presumably microglia cells are not just pruning the visual axons, but also important for pruning all over the CNS during development. So did you see any other changes apart from visual behavioral deficits?
Right. That's a good question. My focus has been on the retinal ganglion cell axon arbors, and presumably there might even be changes at the tectal cell dendrites. There could be like changes at the level of the retina. So like that's all stuff that has yet to be looked at. So that's a good question. But yeah, haven't done anything like that yet.
No, yeah, I was just wondering whether maybe the period you had looked at, it's specifically that during that period, the visual system is being refined, whereas other systems are refined at different time points, or, that perhaps particularly the visual system is somehow impaired, or...
So you're wondering, are you getting at like there's a critical period for refinement? Is that by microglial refinement?
Yeah, that as well, yeah.
Okay. Yeah, that's a good question. Haven't really looked at, like generally in the lab, this is the developmental stage we look at. It's around like 40, developmental stage 43 to 45, or sorry, 47, which corresponds to the tadpoles that you saw, what they look like. At that time, with the behavior test, I can tell you that as the animals are younger, they don't really respond to the looming stimuli as much. So I do think that this is like a critical period for putting together the retinal tectal map, and I think, like I speculate that microglia are important for this process. Yeah, so at least for the wiring. And I do think, yeah, this is a critical period, but I don't know what happens afterwards. So for example, in these animals that have miswired circuits, like if I let the microglia come back, do they fix it? Don't know. Yeah, that's a very interesting question that, again, don't really have any data on that.
Yeah, thank you, Yejing. That is a really important question. There's one more question from Jennifer.
Yeah, actually, it's kind of a little bit related, and you were already talking about this response. And yeah, at first when you said, "Oh yeah, the tadpoles, they aren't responding to the looming stimuli," I'm like, "Oh yeah, you broke the retina." But then you show the reversal. That's really surprising, right? I mean,
yeah, that is really surprising. And that, honestly, like, Ed was the one that was like, "Hey, you should try like the bright looming stimulus." Like, I hadn't even thought of that, and then we tried it, and then we're just like, what's going on? But
oh,
so
you don't know what's going on.
Yeah, I don't really know. I have a model, and I think it's not like, let's see, we can bring it up. It's not like the best model, because it's not. I'm not trying to say that this is like the end-all explanation. It's just a proposed
I mean,
model.
I don't know of any models, so I mean, this is probably
okay. So let me, and Arjun will probably like shoot this down, but like basically the idea is that dark looming, like perhaps during development, you have like the dark looming sensitive retinal ganglion cell neurons wiring together with, like, they should be wiring with the threat detection circuits, but they also may miswire with non-threat detection circuits. And we have also the bright looming sensitive retinal ganglion cell neurons that should be mainly wiring with the non-threat detection circuits, but they may also miswire with the threat detection circuits. And what microglia are doing are they're just getting rid of that miswiring, so that when you activate the dark looming sensitive retinal ganglion cell neurons, you activate the threat detection circuits at the optic tectum, and you elicit escape behavior. So maybe when microglia are depleted, instead, like this miswiring remains, and so that when you activate the bright looming sensitive retinal ganglion cell neurons, that ends up activating the threat detection circuits, and that's why like there's a switch. But this is just a speculation at this point.
It's equally likely to be cadherin mediated, right?
Yeah.
Yeah, I mean, I think this is all super awesome. Yeah, I'd like to talk to y'all a little bit more about this. Yeah, that's cool.
Okay, so just in the interest of time, because we've run out of time, I want to thank everybody for showing up and for great questions. And I just want to do a quick plug for next week's Neuro Light Lunch. So our speakers will be Mark Brandon and Mathilde Balbi. Mark will be talking about imaging the neural basis of cognition 1,000 neurons at a time, so using miniscopes. And Mathilde will be doing multimodal approaches to assess optically induced neuroprotection in acute phase after stroke. So at least in part using mesoscopic imaging. So I hope to see you all back again next week, and thank you for coming. And Tony, let's just thank Tony for a great talk. Sorry to.
Thanks for all coming, and thanks again, Ed, for setting this up. This is great, and Mario, thank you.
Good, and we'll see you all next week. Take care, everyone.
Bye, everyone.
Thank you, Ed. Thank you, everyone.
Thanks, Eve. Good to see you again. Thanks, Mario. All right, I'm going to close this out unless there are urgent questions from anybody. All right, thanks everyone for coming.